Dear students, in this lesson we will be discussing the mechanism of photoperiodism, that is one of the laws of reproduction. So here are the laws. Define photoperiodism: long day, short day, and day neutral plants. Describe mechanism of photoperiodism with respect to mode of action of phytochromes, and then differentiate between floragen and phytochromes. Here we go. Photoperiodism. So from this word photo, you have an idea that photo is something that is related to light. So photoperiodism is a biological response to a change in the proportion of light and dark in a 24 hours daily cycle. Plants use it to measure the seasons and to coordinate seasonal events such as flowering. So photoperiodism is an important phenomena using which the plants have a specific season to flower and to reproduce. So photoperiodism can also be defined as the developmental responses of plants to the relative lengths of light and dark periods. The relative length of day and night is known as photoperiod. Photoperiod is the relative length of day and night. Some plants need to be exposed to sunlight for a particular duration of time to induce flowering. This is the reason behind the seasonal blooming of flowers in a particular species. So there is a well-defined critical period which is different for different plants. Let's discuss what is this critical period. Critical period is the number of hours in the light in a 24 hours day. Some plants flower if dark period is less than a critical period, that are long day plants. And some plants flower if dark period is more than the critical period, that is short day plants. First we will define these classifications. Then we will understand the mechanism. See, based on this duration, plants can be categorized into three major categories. First is long day plants, LDP. Flowering induced by dark period shorter than a critical length. See, here you need to understand as long day plants. They were named because when they were first studied, it was observed that like the plants, they are actually the duration of day is actually what it matters, but later studies showed that it is not the period of light or the duration of day, but it is the duration of night which is responsible for to stimulate the flowering. So long day plants are actually short night plants. We will discuss in the next slide. Short day plants. Short day plants flowering induced by dark period longer than a critical period. We will come to the examples as well. And day neutral plants, plants in which flowering occurs irrespective of day length or night length. So they are like pea, rose, maize, tomatoes. They don't have any particular season, so they don't depend on the critical period. Through this diagram we will understand. See, short day plants, long day plants. Short day plants, they flower when a period of darkness uninterrupted exceeds a critical night length. See, the middle line, the dotted line actually represents a critical length. The dark portion, it represents darker period, and the light portion, it represents daylight, sunlight. So this is critical length. And short day plants need, short day means they need a longer darkness or longer dark period. So this is a critical length. For example, it can be 8 hours, it can be 10 hours. If the darkness exceeds this critical length, see, the darkness is, it exceeds the critical length, then flowering is induced. On the other hand, if the darker period or the period of darkness is less than a critical length. This is the critical length, and the dark period is less than the critical length, then flowering is not going to be stimulated. And here we already mentioned that short day plants flower when period of darkness uninterrupted. So here the period is greater than the critical length, but it is interrupted at different intervals by artificial light having wavelength equivalent to the natural light which was necessary for flowering. So it was the dark period was interrupted, then the flowering is again not induced. On the other hand, long day plants flower when a period of darkness is less than a critical length. See, this is critical length and the darker period is less than the critical length, so they flower in this condition. So they are long day plants because day duration is greater and night duration is less, and night duration is what matters. On the second picture you can see the critical darker period is greater than critical length, so flowering is not induced in this case. And in the last case, if the darker period is greater than the critical length but it is interrupted, so if it is interrupted means the darker period is shortened, it becomes short and shorter than the critical length, and shorter darker period is important for long day plants. So here is a brief history. Photoperiodism was first studied by Garner and Allard 1920 in tobacco plant, where they observed that flowering was induced only after exposure to short days. So later studies indicated that actual stimulus for flowering is uninterrupted darker period rather than light period. Further night interruption experiments have revealed that beside dark period, the quality of light is also necessary to induce the flowering. We will discuss the night interruption experiments and then you will clearly understand this phenomena. So let's move to it is phytochromes, there is misprinted. Photoperiodism depends on light sensitive pigments called phytochromes, which exist in two forms. We will discuss with this picture. Phytochromes are light sensitive pigments which exist in two forms. One is phytochrome red PR or phytochrome 660. It is because of the wavelength which they absorb. And the second is phytochrome FR. FR is for far red. So what happens? Actually, these phytochromes they absorb wavelength of different like phytochrome red, which is also known as phytochrome 660, absorbs red light, and phytochrome FR or phytochrome 730 absorbs far red light, and they are interconvertible. Each can convert to other when they absorb light. If for example, if PR absorbs red light, it is going to be converted into PFR, and if PFR absorbs far red light, it is going to be converted into PR. But see what happens during daytime. Red light and far red light both are present in the visible light, so the cycle continues. They keep converting into each other, like far red in presence of far red light, PR is going to be converted into PFR, and in presence of far red light, this is going to be converted into this one. But the actual thing which is necessary is night time, dark. In dark, what happens? In dark, in the night, red light is not available. So the PR or P660, which was converted into PFR during the day, and it was accumulated in the dark, red light is not available, only far red is available. So this is converted into PR or P660. It converts slowly. But what happens? This PR is actually necessary. It builds up and triggers floragen release in short day plants. You can also remember with the short wavelength. The phytochrome having short wavelength is important for short day plants, and the phytochrome having greater wavelength is necessary for long day plants. You can remember it in this way. So what happens? The phytochromes detect and absorb specific wavelength of light and stimulate production of a hormone, that is floragen, in leaves, which travel to the floral buds and initiate flowering. So phytochromes are the pigments and floragen is the hormone. Phytochromes, the amount of the concentration of phytochrome is important to trigger the or the production of this hormone. So in short day plants, when P660 is in greater concentration, floragen is going to be released, and in long day plants, when P730 is in greater concentration, it is going to stimulate flowering. In this picture you can more clearly understand. See, short day plants require low P730 to 660 ratio, means they need less P70 phytochrome. However, therefore dark period longer than critical period will convert all the accumulated P730 to P660. See, if darker period is greater, all the P730 is gradually going to be converted into 660, and the concentration of P70 will be low as compared to P660. So when this concentration is low, it is if this concentration is low, obviously the concentration of 660 is going to be high, and in short day plants, this high concentration of 660 stimulates floragen hormone. It travels through phloem and induces flowering. But on the other hand, long day plants require high P730. They need more P730, and if darker period is less, it will not be converted into 660, and the concentration of P730 will be greater as compared to P660, and greater concentration of 730 is important in long day plants to stimulate the production of floragen hormone, which will induce the flowering. So what is important, again I'm revising it for you. See, during day red light is more available, so P660 is converted into P730 and it is accumulated in the plants. During night time, if night period is greater, if the dark period is greater, this will maximum of the 730 will be converted into phytochrome 660. So greater darker period means greater P660 and less P730, and less P730 is important for low P730 to P660 ratio is important for short day plants. And if the darker period is short, means in case of long day plants, darker period is short. Short darker period means less of the P730 is going to be converted into 660, and less darker period is favorable for long day plants because the concentration of P730, high concentration of P730 is important for long day plants. I hope this was clear. So here is the night interruption experiments. Again, these experiments show that this middle line is again representing critical period, critical darker period. So on the top of the picture you can see short day plants, and at the bottom you can see long day plants. So what were the experiments? When the darker period is less than the critical period, flowering is induced in long day plants and it is not induced in short day plants. In the second picture, When the darker period is greater than the critical length, flowering is induced in short day plants, not in long day plants. So in the third picture you can see when this darker period is greater than the critical length, but there is an interruption experiments. These are night interruption experiments. There is interruption by an artificial red light. So red light, I already told you that, see, red light is going to make phytochrome 730, and 730 is important for long day plants. So this R represents red light, and red light, if it is interrupted by red light, flowering is stimulated in long day plants. Although the darker period was greater than critical period, but interruption with red light resulted in flowering of long day plants. Next, here you can see there are two types of interruption. First, it was interrupted by red light, then it was interrupted by far red light. So you have to keep in your mind that the interruption which, or the light wavelength of light which was given later matters actually. Initially they gave red light. When in this experiment, when they gave red light, flowering was stimulated in long day plants. But here they gave red light, and after a brief period they again interrupted with far red light, and far red light is actually going to convert P730 into 660, and that is favorable for short day plants. In the next experiment again they interrupted with red light, then far red light, then again red light. So the last interruption matters, and it is favorable for long day plants, and same it is continued with more interruption like red light, then far red light, then red light, then far red light, and the last interruption again matters, and it initiates flowering in short day plants. So this was all about photoperiodism. What is actually photoperiodism? It is the period of light or the period of dark that stimulates the flowering in different plants. So on that basis, plants have been divided into short day plants, long day plants, and neutral plants, day neutral plants. So short day plants are actually long night plants because they need critical period greater than they need a darker period greater than a critical length. Every species has a specific critical length. Coming back to this slide, see, long day plants, henbane is an example, and it needs critical period 13 hours, means flowering is induced by dark period shorter than critical length. This is the critical length. 13 hours is the maximum length, and flowering will be only induced when the darker period is less than 13, 12, 11, 10, etc. So examples of short long day plants are henbane, cabbage, radish, spinach, sugar beet, and etc. You can add more examples. And short day plants, what was the definition? Flowering induced by dark period longer than a critical length. So this is a critical length. For example, cocklebur, critical length is 8.5 hours, and for tobacco is 10 to 11 hours. So in order to induce flowering in these plants, the darker period should be greater than this critical length. It should be 12 hours, it should be 13 hours. So the critical length is different for different species. Here you can see for cocklebur it is 8.5 hours, for tobacco it is 10 to 11 hours. Examples of short day plants are sunflower, rice, soybean, tobacco, strawberry, etc. And day neutral plants, I already mentioned that they are independent of light or the dark period, so they can flower at any season. So this was all about photoperiodism, short day plants and long day plants. So I hope it was good and you must have understood it clearly. Thank you for listening.